Hey there, science fans. Today, we're diving into the fascinating world of lipid bilayer vesicles. Picture this: tiny bubbles, way smaller than the width of a human hair, with the power to revolutionize medicine, food science, and even environmental cleanup. Pretty cool, right? These microscopic marvels are essentially tiny containers made from lipid bilayers, which are like thin sheets of fat molecules. They can encapsulate and transport all sorts of goodies, like drugs, nutrients, or even tiny particles. Think of them as microscopic delivery trucks, carrying their precious cargo to specific destinations within the body or the environment. Why all the excitement about these tiny bubbles? Well, for starters, they hold immense potential for targeted drug delivery. Imagine delivering medication directly to diseased cells, minimizing side effects and maximizing therapeutic benefits. That's the power of vesicles. But wait, there's more. These versatile vesicles are also making waves in areas like food science, where they can protect and deliver sensitive nutrients. And in environmental science, they're being explored for their potential to remove pollutants from water and soil. So buckle up as we explore the amazing world of lipid bilayer vesicles. Before we delve deeper into the fascinating world of vesicles, let's take a closer look at their fundamental building blocks: lipid bilayers. These structures are essential to many biological processes, and understanding them is key to grasping how vesicles function. Remember those mesmerizing soap bubbles you loved to blow as a kid? They floated effortlessly, shimmering in the sunlight, capturing your imagination. Well, lipid bilayers are kind of like those bubbles, but on a much smaller scale and with a slightly more complex structure. They form the basis of cell membranes and are crucial for cellular function. Imagine a thin, flexible sheet made up of two layers of special fat molecules called phospholipids. These molecules are the building blocks of the lipid bilayer. Each phospholipid has a head that loves water, known as hydrophilic, and two tails that try to avoid it, known as hydrophobic. This dual nature is what gives the lipid bilayer its unique properties. When these molecules come together in water, they spontaneously arrange themselves into this bilayer structure. This self-assembly is driven by the hydrophobic effect, where the tails hide from water while the heads interact with it. The hydrophobic tails point inward, away from the water, creating a protected interior, while the hydrophilic heads face outward, interacting with the surrounding aqueous environment. This arrangement is both stable and flexible. This unique arrangement creates a stable barrier that can encapsulate a watery solution inside, forming a vesicle. Vesicles are like tiny bubbles that can carry substances within them, protected by the lipid bilayer. Think of it like a sandwich. The bread slices represent the hydrophilic heads, while the yummy filling represents the hydrophobic tails. This analogy helps visualize how the bilayer is structured. The bread slices, or hydrophilic heads, interact with the watery environment, while the filling, or hydrophobic tails, stay protected inside. This simple yet effective structure is fundamental to many biological processes. And just like a sandwich can hold different fillings, vesicles can encapsulate a variety of substances. They can carry nutrients, waste products, or even medications, making them incredibly versatile tools in both nature and medicine. Pretty neat, huh? Now that we understand the basics of lipid bilayers and their remarkable properties, let's explore how we can harness them to create these incredible vesicles. These tiny structures have vast potential in scientific research and medical applications, opening up new possibilities for innovation and discovery. Creating these microscopic vesicles with precision requires a special set of tools. Enter microfluidics, a technology that manipulates tiny volumes of fluids in channels thinner than a human hair. Think of it like plumbing, but on a miniature scale. Microfluidic devices, often made from materials like glass or polymers, contain tiny channels and chambers where we can precisely mix, react, and manipulate fluids. By carefully controlling the flow of fluids containing our lipid molecules, we can coax them to self-assemble into vesicles with remarkable control over their size and shape. Imagine two streams of water merging in a river. The point where they meet creates a unique environment where our lipid molecules can interact and form vesicles. That's kind of similar to how microfluidics works, but on a much smaller scale and with incredible precision. This level of control is crucial for creating vesicles suitable for various applications, from drug delivery to food science. By tweaking the flow rates, channel dimensions, and lipid compositions, we can customize the size, shape, and properties of our vesicles to fit specific needs. Section 4. Building bubbles with microfluidics, how it works. In this section, we will delve into the fascinating world of microfluidics and explore how we can create tiny vesicles using advanced techniques. So, how do we actually make these vesicles in microfluidic devices? The process might seem complex, but it's quite ingenious and efficient. There are several methods, each with its own advantages. These methods have been developed and refined over the years to ensure precision and control in vesicle formation. Let's explore a couple of popular ones. Understanding these methods will give us insight into the versatility and potential of microfluidic technology. First, we have the meeting of the streams method, also known as microfluidic hydrodynamic focusing. Remember that river analogy? This method is akin to two rivers merging into one, creating a new flow with unique properties. In this method, we use microfluidic channels to inject a stream of lipids dissolved in an organic solvent into a central channel flowing with water. The precise control of these streams is crucial for the formation of vesicles. As the two streams meet, the lipids encounter the water and quickly self-assemble into bilayers, forming vesicles. This self-assembly process is driven by the hydrophobic and hydrophilic interactions of the lipid molecules. The flow rates and channel geometry precisely control the size and uniformity of the vesicles. By adjusting these parameters, scientists can create vesicles of specific sizes tailored for different applications. Next, we have the bubbling up method, or microfluidic droplet generation. This method is particularly interesting because it mimics a simple, everyday activity. This method is like blowing bubbles in your milk. Just as you create bubbles by blowing air into the liquid, we create droplets by manipulating fluids in microfluidic channels. We use microfluidic channels to create tiny droplets of a lipid-containing solution in an immiscible fluid, like oil. These droplets are the initial templates for vesicle formation. These droplets act as templates for vesicle formation. The controlled environment within the microfluidic channels ensures that the droplets are uniform in size and composition. As the solvent in the droplets diffuses into the surrounding fluid, the lipids assemble into bilayers, forming vesicles. This diffusion process is carefully monitored to achieve the desired vesicle characteristics. Again, the droplet size and therefore the vesicle size can be precisely controlled by the flow rates and channel design. This level of control is essential for applications that require vesicles of specific sizes and properties. These methods, along with other innovative techniques, allow us to create vesicles with remarkable precision and control. The ability to fine-tune the vesicle formation process opens up new possibilities in research and industry, paving the way for exciting applications in various fields. From drug delivery systems to synthetic biology, the potential uses of these vesicles are vast and varied. The precision and control offered by microfluidic techniques are revolutionizing how we approach these applications, making them more efficient and effective. Section 5. Size and content control. Fine-tuning our vesicles. Imagine baking a cake. You wouldn't just throw all the ingredients together without measuring, right? You'd carefully measure each ingredient to ensure the cake turns out just right. The same goes for making vesicles. Precision is key. We need precise control over their size and composition to ensure they function effectively for their intended purpose. This precision is crucial for their performance. Microfluidics gives us that control. It allows us to manipulate fluids at a very small scale. By adjusting parameters like flow rates, channel dimensions, and lipid concentrations, we can achieve the desired vesicle characteristics. We can fine-tune the size of our vesicles to meet specific needs. This level of control is essential for various applications. Need tiny vesicles to sneak through tight spaces? No problem. We can create vesicles small enough to navigate the narrowest channels. Want larger ones to carry a bigger payload? We can do that too. Larger vesicles can be designed to transport more substantial materials. we can do that too. The versatility in size allows us to tailor vesicles for different tasks. But it's not just about size. The composition of our vesicles is equally important. The materials we use can significantly impact their functionality. We can incorporate different types of lipids with specific properties into the bilayer, tailoring the vesicles for specific interactions. These lipids can be chosen based on their ability to interact with certain cells or respond to environmental cues, like those that can interact with certain cells, ensuring targeted delivery, or respond to environmental cues, allowing for dynamic responses in different environments. This allows us to customize vesicles for targeted drug delivery, controlled release, and other exciting applications. The potential uses are vast and varied. Think of it like building a house. The size and shape of the house depend on your needs and preferences. The size and shape of the house depend on your needs, and you can choose different materials for the walls, roof, and foundation based on their properties. And you can choose different materials for the walls, roof, and foundation based on their properties. Each material serves a specific purpose. Similarly, we can design and build vesicles with specific sizes, compositions, and properties to suit a wide range of applications. This customization is what makes vesicles so powerful. Similarly, we can design and build vesicles with specific sizes, compositions, and properties to suit a wide range of applications. From drug delivery to diagnostics, the possibilities are endless, and properties to suit a wide range of applications. The versatility of vesicles opens up numerous possibilities in science and medicine.